All right, good morning, everybody. We will get started here in just a moment. All right, hello, everyone, and welcome to today's keynote webinar titled The Extracellular Vesicle Landscape from Biomarker Discovery to Therapeutics Opportunities. This webinar is a part of the 7th annual event in the Cell Biology Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, Paolo Neviani, Director and Founder, Extracellular Vesicle Core Children's Hospital, Los Angeles. For a complete biography on our speaker, please find it on the left of your screen. Paolo, you may now begin your presentation.
Hello, good morning, everyone. Thank you so much for the kind presentation, and I want to thank the organizers for inviting me to give this talk about extracellular vesicles. Let me start my screen share here. Okay, so today I'm going to talk about extracellular vesicles, which is a topic near and dear to my heart, and I'm going to talk about the core, the Share Lab core that is at the Children's Hospital Los Angeles that was founded by myself in 2018, and it was the first core of its kind in the country. Actually, the second in the world after another core that was present in Finland. This is the outline of my presentation today. I will initially introduce extracellular vesicles by giving a definition, talk about biogenesis, functions, and potential therapeutic applications. I will then talk a little bit about my experience with extracellular vesicles at Children's Hospital and the reasoning behind the funding of the core lab, and give an overview of the core services. By presenting current methodologies in EV research in general, and specifically for the ones that we offer at the core lab. And I will conclude with the real world examples of applications that were made possible with the support, with partial support of the core lab. So what are extracellular vesicles? Extracellular vesicles are extracellular particles that are defined by a lipid bilayer. So they have a plasma membrane, and you can see here, this is a pretty generic figure that represents a generic structure of an extracellular vesicle. It has a lipid bilayer and several proteins. They contain a heterogeneous cargo of nucleic acids, such as RNA, microRNAs, DNA, and protein, and other metabolites. They are released from all cell types and are found in all body fluids. There are different types of extracellular vesicles, and we recognize three main categories based on their size, but also based on the biogenesis. The larger extracellular vesicles are the apoptotic bodies, which I will not spend any time to talk about here, because the core lab actually focuses on these two categories, which are microvesicles that are loosely defined as vesicles with the size of 100, between 100 nanometer up to a few microns, and exosomes, which are smaller vesicles. They're also loosely defined as vesicles between about 30 to 150 nanometers. The size, these vesicles do differ in size, but what makes them unique and what really divides them into two different categories is their different biogenesis. It is known that exosomes are of endosomal origin. They are packaged inside the cells in late endosomes by protein complexes called escort, which are able to sort specific proteins, RNA, DNA, and molecules that are then packaged inside endosomes in intraluminal vesicles of the size between 30 to 150 nanometers. These vesicles are then found in these multivesicular bodies, which are late endosomes filled with mature vesicles. They're then secreted by active exocytosis by a mechanism involving Rab proteins and SNOR proteins, which induce fusion with the plasma membrane, and then exosomes are then released in the extracellular milieu. You can see here, this is a genetic structure of an exosomes, which is pretty similar to all type of vesicles. What's different about exosomes is that because of their endosomal origin, they do have very specific components, and they've also been studied more compared to other types of vesicles, and a lot more is known about specific markers that are found on these vesicles. For example, tetraspinins are very common, CD63, CD81, and CD9. These are plasma like membrane proteins that span the membrane 4 times and are generally seen as common exosomal markers. There's also other proteins such as ALEX and TG101 that are part of the export complexes and are of endosomal origin. The other type of vesicles are microvesicles or ectosomes, which differ from exosomes not only because they tend to be larger, but from the plasma membrane. So they do have similarities with exosomes, but their biogenesis is different. They've also been historically less studied, and their content is less defined, and it usually recapitulates the cell of origin. Some markers are known, but so far we do not have any specific marker that can universally recognize these type of vesicles. It is also important to know that while I'm talking about exosomes and I'm talking about microvesicles, it is very difficult by utilizing the current methodologies to specifically isolate exosomes versus microvesicles. And also because it has been found that there are microvesicles that bud from the plasma membrane that are also just as small as exosomes. So it tends to be more accurate to refer to these two categories as small EVs between 30 to 150 nanometer, which includes exosomes and the small microvesicles budding from the plasma membrane, and large EVs over 150 nanometers, which encompass the larger type of vesicles, including the microvesicles and ectosomes. So this is a timeline of the discovery of extracellular vesicles. The presence of vesicles was demonstrated in the 60s and 70s. That's when membrane enclosed vesicles were were started to be described in the literature, and the fact that cells were shedding vesicles. The multivesicular bodies have been described in the early 80s, but the function was not known. And initially, exosomes were coined sort of like as a cell garbage disposal system, and as a way of the cells to dispose of unwanted material. It wasn't until in the late 90s and early 2000s that the suspicion of an actually more specific role and function of EVs came up in the literature. And in 2007, this paper came out in Nature Cell Biology, which was the first but to report that exosomes contain functional mRNA and microRNA molecules. And this RNA is transferred to target cells, and it is functional in this new location. So this was the first proof that exosomes work as intracellular communicator, and not only exosomes, but also microvesicles. So the vesicles released by cells are taken up by, they're released by secreting cells, taken up by recipient cells, where they deliver their message, and the message is actually functional. So mRNA can be translated into a functional protein, or a microRNA, for example, can target its downstream predicted targets. Since that report in the early mid-2000s, a lot of considerable interest has been devoted to the research mainly into exosomes, and the functions has started to be unraveled. Early on, they started studying the angiogenic functions of EVs, and also the biogenesis, for example, Arab protein mediated release came into focus in the 2010s. And because of the considerable interest in understanding the biology and the functions of EVs, the International Society of Extracellular Vesicles was founded in 2011 with the mission of developing guidelines and to standardize research in extracellular vesicles. These guidelines came out in 2014 and in 2018, and have become sort of like a pillar in research surrounding extracellular vesicles. So during this time, so basically in the past 15 years or so, there's been a lot of interest in unraveling the biology of extracellular vesicles at all levels, including uptake and release, which happens in many different ways. Still, there's many different mechanisms that allow uptake and release of the content of the EVs in the target cells. For example, receptor-mediated uptake, which is specific, specified by receptors, or for example, macropinocytosis, phagocytosis, Cavill-independent endocytosis, and endocytosis, and general endocytosis. The biogenesis has also been the focus of a lot of effort into understanding how the EVs are packaged inside endosome in multivesicular body and released by diffusion with the plasma membrane. But I would say, arguably more importantly, there's been a lot of effort into understanding the content of extracellular vesicles in the effort to elucidate its functions and to potentially develop therapeutic applications. So there's the plethora of functions and applications that have emerged in the past decade or so, and these are generally dictated by the local and systemic delivery of functional RNA or protein or other molecules to the target cells and to their environment. Generally speaking, regarding biological functions of EVs, they tend to be regarded as cell-to-cell communicator or cell-to-environment communicators. By inducing for example, metabolic reprogramming of cells, they can modulate the microenvironment by, for example, modulating the extracellular matrix, modulate the activity of immune cells, induce angiogenesis and also dictate organotropism in metastasis. But I'll talk a little bit more about these concepts here. So the crosstalk between cells is a very important function of extracellular vesicles, particularly in the tumor microenvironment. This is a two-way highway of communication between cancer cells and the tumor microenvironment, in which cancer cells release tumor-derived EVs that are taken up by, by the tumor microenvironment. They may be taken up by resident immune cells, or they may have activity on the extracellular matrix, resulting in, for example, alteration of tight junctions, production of pro-inflammatory cytokines, generation of pre-metastatic niche, alteration of extracellular matrix, and stimulation of angiogenesis. Interestingly, when immune cells or other cell types in the tumor microenvironment, which are not malignant, do take up cancer-derived exosomes, these cells are then sort of educated and modified by these EVs. They can then release their own EVs that can also be altered and influence the cancer cell behavior themselves. So microenvironment-derived exosomes, for example, when produced by educated cells of the microenvironment, can enhance growth and survival. They may induce invasion or epithelium-mesenchymal transition, and overall induce drug resistance. So this is a very important concept where we can tell that, for example, for eradication of cancer cells, we do not. It's probably best to not focus only on killing the cancer cells, but also to study the effect that the cancer cells has on their surrounding, based on this type of communications. So, for example, this is a list of some microRNAs that have been identified in the alteration across talk between cancer cells and tumor microenvironment. And we see several microRNAs that are shuttled between, for example, lung cancer cells, neuroblastoma cells, breast cancers, into the surroundings, like tumor-associated macrophages or endothelial cells, and this goes both ways. Just for this, for here, the take-home message is really just that the shuttling of EV microRNA between cancer cells and surrounding cells promotes a pro-tumorigenic and drug-resistant microenvironment. EVs also have a profound role in angiogenesis, which is one of the first functions that has been identified. They tend to elicit pro-angiogenic signals based on their contact and the surface molecule expression by transferring nucleic acid like RNA, microRNAs, long non-coding RNAs, and circular RNAs, or transfer of proteins such as VEGF, activation of signaling pathways in the target cells, which are usually endothelial cells, or transfer of transcription factor. The role of EVs in promoting angiogenesis is it's a particular interest in tumorigenesis, in which researchers have seen that localized small tumor can release exosomes that are then going to influence and alter the metabolism of the endothelial cells and induce pro-angiogenesis and promote vascularization of the primary tumor. So very important in the tumor progression stages. EVs are also important in metastatic disease and in determining the fate of where metastasis will develop. In the metastatic niche formation, there's what we call a seed and soil model, where the exosome can be considered a fertilizer, in the sense that primary tumor release exosomes that do go in circulation and find a secondary tumor target in specific organs. Here, they can induce many changes, such as upregulation of inflammatory molecules, immune suppression, increased angiogenesis, and remodeling of extracellular matrix. These are all changes that create a permissive state in our target organ that is then permissive for the arrival and for the arrival of the circulating tumor cells, where they can then develop metastasis. Organotropism is also very important when it comes to tumor-derived EVs. That is usually dictated by the presence of integrins on their surface, and the EVs are then able to find specific organs, and that will be permissive for the development of metastasis. After showing all of these functions, we can talk a little bit about the potential therapeutic application of EVs. There's three main categories that mostly come to mind when we talk about therapeutic applications. Which are biomarkers, as diagnostic and prognostic biomarkers in liquid biopsies, drug delivery as a natural alternative to nanoparticles, and finally, regenerative medicine. Regarding EVs as biomarkers, due to their ubiquitous presence in different body fluids, exosomes and in general EVs are considered as potential biomarkers, which have the advantage of being, found in different body fluids. So the concept of liquid biopsy that is very, easily accessible for early diagnosis, prognosis, and monitoring the response to therapy. All types of cargo of EVs is being investigated in this regard, and several microRNAs, for example, have been found as being potential biomarkers for all types of solid tumors that can be found in different sources, such as urine, serum, plasma, or CSF, or other more easily accessible, relatively easily accessible, organs. Other potential biomarkers are exosomal long non-coding RNAs. For example, here they found the signature of seven long non-coding RNAs that can be used as blood-based biomarkers for gastric cancer, or exosomal proteins. For example, Claudin 4, which is a tight junction protein, has been described as a potential biomarker for ovarian cancer. And more recently, the specific subpopulation of EBs expressed in two different theta spanning is also being described by actually one of our collaborators as a potential biomarker for retinoblastoma, which is detectable in August tumor. But I will actually talk a little bit about this more later in the presentation. Currently, there's only one test that has been approved, which has been developed by the company Exosome Diagnostics, which was the first to develop an exosome-based urine biomarker test for prostate cancer patients. The company found that they found an exosomal RNA signature of 3 genes, which is used to generate a score to help clinicians decide whether biopsies of the primary tumor are necessary. And this was granted FDA breakthrough designation in June 2019. Regarding therapeutic applications and in the concept of utilizing EVs as a therapeutic agent, a lot of interest has been given to stem cell-derived EVs, particularly mesenchymal stem cell-derived EVs. This is because it has been discovered that in many cases, EVs are derived from cells. Stem cells have the same activity as the stem cells themselves, but they offer the advantage of offering a type of therapy which is cell-free with no risk of malignant transformation and also low immunogenicity. In general, we see that EVs have stem cell-derived EVs have anti-inflammatory effects. They promote tissue regeneration, decrease fibrosis, for example. They can be rapidly distributed to the injury site since they're a lot smaller than natural cells. And also they can cross the blood-brain barrier, which is a very important function of EVs, which can induce neuroprotection in neurological diseases. There's a lot of preclinical studies in the effort of developing therapeutics which are derived from stem cells and all different type of diseases. I'm just gonna give a few examples here. For example, in the liver, they found that exosomes from human umbilical cord blood mesenchymal stem cells ameliorates liver fibrosis by inhibiting epithelial mesenchymal transition and of hepatocytes and promoting liver regeneration. At kidney level, for kidney disease, actually one of my colleagues here at Children's Hospital is studying amniotic fluid-derived stem cells. Dr. Perrin studying these cells, and particularly their EVs. And they found that AFC-derived EVs prevent glomerular endothelial cell damage in Alport syndrome, which is a chronic kidney condition, by capturing excess VEGF. Another example, in the brain, the group have found that exosomes from human bone marrow mesenchymal stem cells improve cognition and memory impairment, reduce plaque deposition, and beta amyloid in the brain of mice. So has a potentially promising intervention for Alzheimer's disease. Lastly, another potential application of EVs in therapy is drug delivery. This is because they pretty much look like nanoparticles, but they're natural, but a natural alternative to nanoparticles, and they're promising because of their low immunogenicity, their high stability, and also the high delivery efficiency and bioavailability. There are several preclinical studies, for example, of exosomes loaded with curcumin or loading with other chemotherapeutic agents such as doxorubicin or paclitaxel that show higher intracellular delivery efficiency and lower levels of toxicity because the chemotherapeutic agents is delivered directly into the cells. Another application is, for example, utilizing EVs for the delivery of siRNA, for RNA-based gene therapy. In general, EVs can be engineered, they can be loaded with different components, and there's a couple there's a few different ways to do that, either by preloading the packaging cells. So, for example, transfecting cells with plasmid or microRNA or drug that is then packaged inside naturally packaged inside the EVs, or post-loading, in which an active compound is directly transfected into the EVs by utilizing transfection reagents, by electroporation, incubation, conication, or other techniques that have been in development. So far, there really aren't any approved therapeutic application, but there are several clinical trials ongoing and at different phases. This is just an example of a few of clinical trials that are ongoing. See here, a lot of these are from exosomes derived from mesenchymal stem cells. And for example, this one for pancreatic adenocarcinoma is delivering an siRNA anti-specific mutant of KRAS. So now I'm going to switch gear and talk about my own experience with EVs and about the core that was founded at Children's Hospital in Los Angeles. So I started working in this field in 2014 when I joined a group of Dr. Muller Fabbri here at CHLA. And we were looking at dissecting the crosstalk between cancer cells, in particular neuroblastoma cells, and the tumor microenvironment. We had a couple of publications during that time. This first one by Dr. Chalakondla was an analysis of the interplay, the crosstalk between neuroblastoma and tumor-associated macrophages. And we found that neuroblastoma cells secrete high levels of MIR-21, which is considered a non-comer, and secreted into their exosomes. These exosomes are then taken up by the tumor-associated macrophages. The MIR is released in the endosomal compartment, where it finds the RNA sensing receptor TLR8. This induces an inflammatory response, an inflammatory and pro-tumorigenic response, which also results in expression of high expression of MIR-155. This MIR-155, another very well-known oncomer, is packaged in these educated tumor-associated macrophages which are modified by the neuroblastoma. It's then shuttled back to the neuroblastoma, where MIR-155 can then function and target one of its natural targets, which is the inhibitor of the telomerase SIRF1, which then results in increased drug resistance. And this other publication that came out in 2019 in Cancer Research, we were looking at the interplay between neuroblastoma, between high-risk neuroblastoma and NK cells in the tumor microenvironment, where it was known that in high-risk neuroblastoma, which is characterized by amplification of MYC-N, MIR-186, which is a known tumor suppressor, its expression is lower. And also TGF-beta is highly expressed in the tumor and found at high quantities in the tumor microenvironment. This results in inhibition of inactivation of the TGF-beta pathway in the NK cells, which results in an inhibition of their cytotoxic activity. An inhibition of their cytotoxic activity, which is a known immune escape mechanisms. We tried then to restore MIR-186 expression, not directly into the neuroblastoma cells, but into the NK cells, because we knew that MIR-186 had was predicted to target the TGF-beta pathway, but also the MYC-N and its partner aurora kinase A. We utilized nanoparticles to transfect MIR-186, and we found that MIR-186 was then found in the NK-derived exosomes, which, so this mechanism was working basically on two different levels. On one side, we were able to, MIR-186 would inhibit the expression of the TGF-beta receptors, so rendering the NK cells immune to the TGF-beta, so they were not inhibited. So their cytotoxicity could be enhanced. But also we found that MIR-186 was packaged into the NK exosomes, transferred to the neuroblastoma, delivered, and it could downregulate MYC and aurora kinase, resulting in inhibition of growth and migration. This is another example of how, by targeting or modify the way that cancer cells and immune cells talk to each other, we could, for example, induce an anti-tumoral effect. So at this stage, this was 2018, Dr. Fabbri was about to actually move on and leave from CHLA, and I had the thought of starting the Extracellular Vesicle Core. The reason, because we had several researchers that were collaborating with Dr. Fabbri. There was a lot of growing interest in the research community. The field was really taking off, and we had some instrumentation available and expertise that could help other researchers to address technical challenges in EV research. So we initially focused on isolation methods, characterization of size and concentration of the EVs, which are too small to be seen by standard microscopy, and most flow cytometer systems available at the time. So there was a need of dedicated instruments, and also at that time, the International Society of Extracellular Vesicle was starting to develop very detailed, non-binding, but very detailed guidelines, which were published in 2014 initially and then updated in 2018. This is a very busy slide. I don't expect anyone to be able to read this, but I just want to show how in 2014, the ISEP was, had a few guidelines, which then ballooned to much more guidelines in 2018, and now we're expecting these guidelines to be updated here later this year or probably next year. So the core services here at the CHLA, they focus on two main areas: the isolation of EVs in an effort to optimize yield, purity, and reproducibility of EV isolates, and characterization in order to characterize size, concentration, and also the composition, for example, for biomarker studies. I'm just gonna go over a few of these methodologies, talking first about isolation methods. So as I mentioned previously, there is no gold standard. There isn't a one-size-fits-all for isolation of EVs, and we do need to choose the type of method we want to use based on the type of sample and the complexity of the sample that we're working with. Generally, we need to take into account two different parameters: size, as you know by now, the size is what characterizes one of the main characteristics of extracellular vesicles, and also the density. The reason being that EVs have been shown to have a very specific density of around 1.15 grams per ml, and in certain types of samples, particularly more complex samples such as plasma and serum, there's a lot of different types of particles. We not only have EVs, but we have a plethora of lipoproteins that range in size and density. So for example, the very low density lipoproteins and the chylomicrons have a density that is much lower than EVs, but they have the same size. HDLs, which are definitely a lot smaller, around 10 nanometers than EVs, are also overlapping in density. So in generally, size-based separation methods can be sufficient for cell culture experiments, for a condition when cells are kept in culture, and we can then collect the condition medium and isolate the EVs based on size. But if purity is an issue, especially in plasma serum samples, then we need multiple sequential isolation steps, which I will be showing right now. So we have there are different methodologies for isolation of EVs with each one with its own advantages and disadvantages. These two, density gradient and size exclusion chromatography, are the ones that are generally utilized most frequently at the core. But there's those other ones such as differential ultracentrifugation on immunoaffinity magnetic sorting and polymer-based precipitation, which are used in very specific cases. Just to give an overview of these methods, differential ultracentrifugation was basically the first protocol to have been developed for the isolation of exosomes. It's probably the most used method, at least until recently, which is pretty simple. It does require access to an ultracentrifuge, but it is lengthy. The recovery tends to be pretty low, and the EVs may be damaged by the shear force. Overall, this protocol requires a sequential centrifugations at at higher speeds, particularly after a 10,000 G a spin of 30 minutes, we get large EVs or microvesicles, and after 100,000 G spin of at least an hour, we get the small EVs. The method of choice that we utilize at the core is actually the size exclusion chromatography, which is done on this pretty simple chromatography instrument called ACTA. This is the method that gives the highest degree of purity based on size. It is gentle, it's scalable, and tends to be more reproducible, but it may, in certain cases, it may not give the best purity. For example, lipoproteins are not separated if we were to just do size exclusion chromatography with a plasma sample. Here, the overview of the methods. A sample is loaded on top of a column that is packed with a matrix or a gel. The sample is then separated by low pressure or even by gravity flow, and we know that the first fractions to come out of the column in the void volume are EV, and then soluble proteins, which tend to be smaller, are then separated later on. Here if we track the content of vesicles in each fraction, we see that fractions 5, 6, for example, have the highest number of vesicles, whereas the protein content is higher in the later fractions, suggesting a good separation between EVs and soluble factors. The density gradient is another method that requires centrifugation. This is a method that gives the highest degree of purity based on density. It tends to be very lengthy, over 16 hours. But it definitely has the highest purity. This is what we normally use for plasma samples, which is loaded on top of a gradient of iodixinol centrifuge for 16 hours overnight, 100,000 Gs. And then we see at the end of the centrifugation that we have two bands, one a low density band containing VLDLs, LDLs, chylomicrons, and a high density band at the density of exosomes, which contains small EVs and HDLs, which can then be further processed by size exclusion chromatography to separate the HDLs, which are a lot smaller than EVs. In the end, we get a product by combining density graded and size exclusion, which is devoid of ApoB, which is a marker for LDLs, and only has exosomal markers. In this case, we're looking at CD81. We then move on to the characterization of EVs. There's different types of characterization, bulk versus single vesicle characterization. Bulk characterization that we do is a pretty standard immunoblotting procedure by following ICEV guidelines, in which this method is useful to show global composition and purity, so to show absence of contaminants. There's different categories, such as surface markers like CD81, which is one of the tetraspanins, extracellular proteins like fibronectin, which has been shown to be specifically associated to EVs, but also found in later fractions. Cytosolic protein of endosomal origin that prove the presence of endosomal proteins, therefore the presence of actual exosomes of endosomal origin. And we then want to check other co-isolates that, such as calnexin, which is an intracellular protein, which we do not want to see in our isolates. So this is just another measure of purity. An important technique for a single vesicle characterization is the nanoparticle tracking analysis. This is the first instrument that we had at the core. This is a dedicated instrument that allows the visualization of exosomes which are too small to be visualized by standard microscopy. The NanoSight is a light scattering and image processing instrument which measures concentration and size distribution of particles in suspension in the size range of 30 to 2,000 nanometer. So the particles are seen as light points moving under Brownian motion. So, and their speed directly correlates with the size. So the instrument, it's able to track single vesicles and to give us a nice size profile and also a concentration. The disadvantage of this technique is that it's non-specific. So we're just looking at particles that are flowing in the instrument. We cannot use specific markers. Newer instruments can do that by fluorescence, but I don't think they're very accurate. If we do want to look at markers, we have this instrument, the ExoView R100, which is based on it's an imaging instrument based on interferometric reflectance imaging sensor, where the sizing is based on the controls. This is an immunocapture-based assay where chips are coated with antibodies that are specific for the tetraspanines. So the EVs are captured. This basically is a combination of isolation and characterization. It does not require pre-isolation because isolation happens as we characterize the vesicles. The vesicles are captured on the chips and counterstained with specific antibodies. And this allows us to see subpopulation of EVs. Here you can see typical results where we see on a spot, for example, this is the CA1 spot. We're looking at single vesicles that are expressing the CA1 markers and other markers. And this is very interesting because it's opening a new era of the ability to see, subpopulations and subpopulation of markers on single vesicles. And it's very important very useful for biomarker studies because these markers can also be swapped for other ones, for example, for biomarkers or cancer markers, and to analyze EVs as the potential biomarkers. There's other techniques that are not available to core now, but are pretty common. Hopefully we can add them on sooner rather than later. Electron microscopy is the only technique that really gives us a more direct visualization of EVs by transmission electron microscopy or cryo-EM. Super resolution microscopy, which is, we actually hopefully are going to add this very soon to the core. This one is an optical technique that goes beyond the diffraction of light. So it's an indirect visualization with fluorescent tags, but allows a limit of detection of about 20 nanometers. So we can actually start seeing the structure in a type of instrument that's a lot cheaper than an electron microscopy. Finally, nanoflow cytometry. There are newer instruments that have a limit of detection of about 40 nanometers or less. So now, it's just in the past few years, that flow cytometry is becoming a lot more and more common and a lot more accessible for the detection of EVs. I'm just gonna spend a few minutes here. I'm almost finished. We're gonna talk about some real-world examples of publications that have been made possible with the support of the core. Here at CHLA, Dr. Kubrinik is studying osteosarcoma, and they found that in the serum of osteosarcoma patients, they found some DNA repetitive elements to be represented. These are the 4 types of DNA repetitive elements, and they found them to be associated with EVs. And interestingly, these are being found to be specifically associated with EVs, but also still sensitive to DNA, suggesting that they may be linked to the vesicles on the outside, which is a very interesting concept. Our colleagues, Dr. Berry, Dr. Shu, the ophthalmology department, they're studying the aqueous humor from pediatric ocular diseases. This is a not so easily accessible aqueous humor. It is a not so easily accessible type of sample, especially because the volume that can be extracted is very small. And previously, the only way to detect EVs was to pull multiple samples. But with the addition of the ExoView instrument at the core, they were able for the first time to see extracellular vesicles on single patients just by spotting just a few microliters of the sample. Here they look by running these different types of samples, they found that in non-malignant diseases, there is a distinct pattern of tetraspanning expression. Particularly, they found that there's a mono CD63 population here shown in green that seems to be a potential eye-specific biomarker. They then looked at retinoblastoma, so a malignant type of disease, and they found that the pattern of expression was completely altered, suggesting that perhaps these EVs are made by the tumor. They then expanded the retinoblastoma research into to 37 different patients. They 10 diagnosis and 27 during treatment, and they found in this newer, more recent publication that double positive CD63 plus, CD81 plus small EV population is higher here, is higher diagnosis than during the treatment and correlates with the tumor burden. Whereas the mono CD63 population is higher during the treatment, so suggesting that a ratio between these two populations may be a candidate biomarker for disease burden and response to treatment. This is very important because the primary tumor, retinoblastoma, cannot be biopsied, so the aqueous tumor can be more easily accessible source of information for the diagnosis and the treatment, so potentially very useful biomarkers. Here, Dr. Green at Aarhus University in Denmark is studying the EVs derived from cancer cells exposed to microgravity. And they found, here we ran ex vivo experiments, we found that more EVs are secreted in microgravity conditions and with an expansion of the CD63 positive compartment, suggesting that EVs could potentially be used as a biomarker of the effect of microgravity on human physiology for space exploration. Finally, I just want to mention this paper that just came out by Dr. de Klerk at CHLA. Here they study organotropism of tumor-derived EVs, where tumor-derived EVs that are GFP positive, GFP tagged, these are captured in the premetastatic niche at the lung and liver level in an organotropic specific manner, and they induce inflammation. So these are GFP positive EVs that have been taken up by mouse. This is in a mouse model, but so by the macrophages. I'm not going to talk too much about this because this is the subject of actual presentation of my colleague and friend Laurence Sart, who will be presenting this, and I believe the presentation is already present on demand during that was published to date for this conference. So make sure that you go learn about organotropism of EVs in neuroblastoma and melanoma in this newer report. So I'm gonna stop here. Here's the website for the core in case you're interested in looking at the core, the services that we do offer. Feel free to ask any questions, and feel free to contact me if you need any questions or any help or protocols for your if you have any interest in starting doing your own research in extracellular vesicles. Thank you very much, and I'm gonna stop here. Thanks.
All right, thank you very much, Paolo, for your informative presentation. We will now start the Q&A portion of the webinar. If you happen to have a question you would like to ask, please do so now. Just submit it in the Ask a Question box located towards the bottom of your screen. Let's go ahead and get started. Are there any isolation methods that do not require specialized equipment? What about precipitation methods?
Okay, yeah, sure. So, yeah, I've been talking a lot about I've been in the need of specific, high-tech equipment, but there are ways to do this. Not, for example, when we do the size exclusion chromatography, we do at the core with the chromatography instrument, but initially we set up this this assay. Basically, it's manually done. We can just manually pack columns and do it just by gravity flow with a very simple fraction, excuse me, fraction collector, or basically just by getting the fractions. So chromatography instruments are not necessary, and there's also companies that do sell columns that are ready to be utilized even without those instruments. Regarding precipitation, precipitation is definitely not my favorite. I don't usually do that. We have shown that precipitation does precipitate a lot of non-vesicular component, and actually early on with the reagents available at the time, about 90% or more of the protein recovery was not actually associated with EVs. But I know that there are newer reagents that I haven't had a chance to test, so maybe the newer ones are maybe better. But I would still recommend size exclusion chromatography if possible.
Perfect. All right, thank you. Here's another. Is there any research on tumor-derived exosomes and immune checkpoint inhibitor resistance in solid tumor malignancies?
Sure. Yeah, this isn't necessarily my expertise, but I know that immune checkpoint inhibitors, there's a lot of interest in PD-L1 inhibitors, and it has been found that exosomes do carry PD-L1. So this is there's definitely a lot of interest in that, and it's an area of exploration for anti-cancer therapy.
All right, perfect. Thank you. It looks like we have time for one final question here. Can extracellular vesicles be isolated from solid tissues? What is the recommended procedure?
Yes, we do that. The methodology is very similar to what we do with all types of samples. We tend to do size exclusion chromatography, and in this case, because it's more of a complex type of tissue sample, we do use a density gradient. What I would recommend is that it's very important when the dissociation of the tissue happens to be very gentle, to avoid the rupture of the cells and the leakage of the intercellular content. So I would recommend a gentle enzymatic dissociation of the tissue with, for example, collagenase, and followed by the normal size exclusion chromatography and density gradient, and it works pretty well.
All right, thank you so much. Thank you again, Paolo, for your informative presentation, your time today, and for your important research. Before we go, I would like to thank the audience for joining us today. Any questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 20th, 2025. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you. Bye-bye.